Hello all, welcome back. Friends, we have started with water pollution and today, as I told you, we are going to discuss on an important factor and that important factor is called as the standard methods, okay, or we can say it as treatment methods. Now, before starting on to a treatment method, let's understand how the water quality or water standard is being measured. Now this water standard is called as the BIS standard method and today we are going to look forward for this BIS standard method. Of course this is not very much crucial for you people, so we will try and understand it in a very brief form, but of course we will try for it. And the next thing which we are going to look forward is what is the treatment for drinking water. If the drinking water, that means whatever the drinking water we are consuming, it is having a method, a typical method, and we have to study this typical method, that is drinking water method. So if you remember, I have told you in early lecture also that every water has a different treatment system. That means if you are talking something about a water which you are drinking, it will have a different water treatment system. If it is sewage water, it will have a different part. If you are talking something about with regards to the system of water which is being used for industries or agriculture or maybe any construction property, it will have a very different system for a usage or a treatment method. Let's understand today the very first part, that is what is meant by the BIS system. Okay. Now this is a chart, like I told you in the last lecture also that it will be completely a chart based study, and of course you do not have to mug up this chart. Okay, so do not try and mug up this chart. It is just for your reference so that you people can understand the exact method what I am trying to tell you, that what is the BIS method. Now again one more thing, important part, that this BIS system is that means whatever the ratios are being provided to you, what are the numerics being provided to you. So you can see I'm giving you two different images over here. That means this is a continued image of the first part. So you can find that we are being given in a chart formation, that is characteristics. That means we have to it's like a chemical reaction what you do in your chemistry laboratories. That means what is the requirement if you are testing a particular kind of a water, what is required or what human body or that particular region will be accepting it, then what is the limit, lower limit, we can call it as lower limit or relaxation limit in short, and what a remark is being covered out. That means what are the effects if it takes place, what will be the exact measure to be taken. For example, in one of the system like this, you can find if the iron, okay, is present, that means acceptable is 0.3 only. So you can find it is very minimum amount which is acceptable, but if it exceeds more than that, it is not acceptable. That means there is no relaxation. Okay, if the water content has more than at least point also more than 0.3, it will not be accepted. That means there are methods in through which this particular chemical product metal has to be removed out and then the concentration will be maintained. Okay, so that is the part which BIS does. Now let's understand what do you mean by BIS first. So BIS is working in each and every country, and the BIS is called in our country, and the BIS is called as Bureau of Indian Standard. Okay, so mark this acronym or note down this acronym, that is it is Bureau of Indian Standard. So it is specific, that means it is one of the you can say it is one of the form of company, or it is one of the form of a norm which the government has put forward to specify something about the drinking water quality, to measure the drinking water quality standard, so that it can be provided to the people of the India for the sole purpose of the drinking. Okay, so it is basically used more for, you can say, for a drinking water. Now it is being tested regularly at a particular intervals of time. It is being tested, and as I said, that it depends completely on the different forms. Like if you are at a particular area, it will be different. So depending upon the region, the type of a water quality also changes, and depending upon the same quality of the water, this BIS standards also varies from region to region, but not at a larger amount. So it is giving us a complete table which can be adapted by each and every part of a country. This BIS is working in our country in 13 different cities, okay, and it is working basically from a point of view or idea that you have to create water which is safe for the utilization. So this is working in 13 countries, and it is working on the idea that the water which is used or consumed by the human will be safe for their health. So the 13 countries are having some major countries. That means we are having some major countries which are carrying out this samplation, water samplation, and it major, you can call it as some metropolitan cities are also included. Are so it is Delhi, Kolkata, Chennai, Bangalore, Jaipur, and Lucknow. Of course, Mumbai is also a part of it, and we specify it on the basis of a number. That means parameter is specified and it is given number. Like we have ISO number, similarly, or ADMARC number, we have similarly the water system number, which is called as water quality number, and it is called as IS number. So IS means Indian standard. So depending upon the quality of the water of the India or a particular region in our country, it is being standardized as IS standard. So IS 10500 2012 is being granted as the IS number for this BIS system. Now what does the parameter tells us? The parameter test basically something called as the pH values, turbidity, what amount of water is turbid, what is the pH of the water. That means you can, if you know that the acceptable limit, whatever we are drinking, the pH should be of around 6.5. Okay, it should not exceed more than 8.5 also, and you do not have any relaxation for this pH. So if there is a large amount of soil, metallic or chemical products which are harmful, of course the pH will increase, and that is not acceptable. So the pH value should be from 6.5 to 8.5. So pH values are being tested. Turbidity is tested. Turbidity in a sense means what amount of clotting is there, what amount of pollutant are present in these. Now of course the pollutant do not consider as hard pollutant. Of course these are the chemical pollutants also which are dissolved in the water, and it is being tested out. Also the E. coli is being tested. Okay, so why to test E. coli? Because it is of course one of the natural bacteria. It is thermobacteria, and it can cause a huge effect on the human health, because if E. coli unknowingly enters into your body, harmful product, a harmful bacterial enters into your body, it will cause the problems like jaundice, cholera, and diarrhea also. Okay. At the same time, taste, odor is also tested, so that each and everything is being tested in such a proper format, so it becomes easy that water is being then provided to the human for the human consumption. Okay, so why are we getting this kind of safe water? Is just because of the BIS system, and one of the method which is being again advised to us is that we use water purifiers. So if you want more protection from these harmful pollutant, now there is a system called as RO water system. Even though the RO purifiers are being used in each and every houses, still whatever the water is being provided to us is completely BIS standardized water, which is then obtained from the system or the government system to us. Okay, so that much is enough from the point of view for you people to understand what is the BIS system. So you should always keep in mind that some major points, for example, the taste, turbidity, pH meters, different chemicals are being tested. Some measures are always there, that particular relaxation, in some cases particular relaxation is provided, and in other cases a relaxation cannot be provided. Especially when it is going for the E. coli test and the pH test, no relaxation will be provided, of course. So if the E. coli exists, that means the bacteria is there, and of course it will not be a useful or drinking form of a water which can be used by the humans. Now, as I said, we are going to see today an important factor that is called as water treatment systems. So this is a very first water treatment system, and this water treatment system is something related to the drinking water system. Okay, so we are going to study today something about drinking water system. And of course, we are going to look forward at this drinking water plant from the point of view of surface water. That means whatever the water is being present on the surface, like rivers, lakes or canals, this water needs to be purified, right, before the use, and this purification of water is done by this method which I am trying to present to you. So you people can just have a look at this particular diagram, and then for your understanding, okay, you can do one more thing, that you can just pause the video over here on this diagram, and I'll just try to make explanation so that you can understand it very clearly from a diagrammatic point of view. Or else you can also do one more thing, that you can just again forward it back, reverse it back, so that it becomes clear for you people to understand what we are trying to understand. Okay. Now, one more thing which is important when we are starting something about this water treatment, that water treatment does not have a signified method. That means you cannot use one method. Like I've said, one method cannot be used for cleaning up the water or purification of water when you are having a different qualities of water. Like if it is drinking water, you have to use different. You have sewage water, you have to use different method. But the same also applies when it is only a drinking water, because from region to region the quality of water also varies. So you can find more amount of salinity in the region of drinking water where it is ocean areas. Okay, so there you have to imply something a different part. When you are talking something about a higher hilly area, especially the northern part, of course there will be a different part, a different system of purification method for the drinking water. So always keep in mind that the process of drinking water, even though we are trying to study, depends upon what kind of region, because region decides the quality of water. And of course, this is done with the help of BIS. Then the municipal corporation tries to clear out the water with the help of water treatment, all the pollutant. And now we are going to try and understand what are the methods of clearing out this pollutant. So for your reference, which can be again very much in a concise form, because I know this is very heavy diagrammatic method which you will sometimes may feel very hampered to understand, but we will try to understand it in a form with the method of this small system. This might not be very clear to you people, but I'll try to make and understand this system. So the very system which is being adopted by the municipal corporation, which is probably used all over the country in our country, is this system. That means surface water especially. Now the surface water basically goes into methods, different kinds of methods, and this different kind of method then finally gives us out a product which is drinkable. Okay, so this drinkable method system includes the processes such as, first is pre-chlorination. Now what do you mean by pre-chlorination? That is, chlorine is being added into the water which is being collected from the rivers, lakes or the ponds or canals. Why it is being done? This is being done to control the development of or growth of algae and biological organisms which are not visible to your eyes. So chlorine is added into the water so that the control of biological growth is done. Okay, so that is the first method. The second method, that means after the pre-chlorination, the second method is followed, that is called as aeration. Now aeration is done for removing out the harmful products such as iron and manganese. So if you remember the BIS chart, we have seen that in the BIS chart you can find that iron, if it is present at 0.3 only, it will be acceptable. If it exceeds more than point also, there is no relaxation. So similar goes with the product that is called as aeration. So what aeration does, after pre-chlorination, आप जब वाटर में क्लोरीन ऐड कर दोगे तो आपका काम क्या करोगे, आप लोग क्या कर रहे हो, दैट मीन्स म्युनिसिपल कॉर्पोरेशन क्या कर रहा है, पानी में पहले क्लोरीन ऐड किया जाता है कुछ अमाउंट में, सो दैट कोई भी बैक्टीरिया या फिर बायोलॉजिकल ऑर्गेनिज्म अगर वाटर में होता है तो उसका ग्रोथ बंद हो जाता है. सेकेंड होता है एरेशन, दैट मीन्स क्या करते हैं हम, सारा अमाउंट जो भी प्रेजेंट होगा, दैट मीन्स आयरन एंड मैंगनीज जो भी प्रेजेंट होंगे उसे एक्सट्रैक्ट आउट करने की कोशिश करते हैं या फिर रिमूव आउट करने की कोशिश करते हैं विद द हेल्प ऑफ एरेशन मेथड. ये कब किया जाता है, प्री क्लोरिनेशन के बाद में किया जाता है. द नेक्स्ट मेथड इज कॉल्ड एज कोएगुलेशन. ओके, कोएगुलेशन में फ्लॉक्यूलेशन किया जाता है या फिर फ्लॉक क्रिएट किया जाता है. नाउ यू कैन अंडरस्टैंड दिस वेरी वेल विद द हेल्प ऑफ दिस डायग्राम. व्हाट डू यू मीन व्हाट इज द मेथड ऑफ फ्लॉक्यूलेशन और what is the term of flocculation? Consider this container is filled with the water, and of course the red dots which you can see is nothing else but the pollutant, okay, or suspended polluted material. Now whenever you are adding a coagulant to it, that means you are trying to add some kind of chemical material which will completely remove your pollutant. And how is this removal taken place? After you add a coagulant to this water container, it will then go into a process of centrifugation. That means consider कीजिए कि हमने एक प्रोडक्ट मिक्स किया है वाटर में जो कि एक कोएगुलेंट की तरह काम करेगा. दैट मीन्स ये सारे पोल्यूटेंट को अट्रैक्ट करके खुद में पैक कर लेगा. सो दैट इज द प्रॉपर्टी ऑफ कोएगुलेंट, दैट इट ट्रैप्स एंड देन completely separates water pollutant from the water. ये ट्रैप करेगा पूरी तरह से वाटर बॉडीज को, एट द सेम टाइम केमिकल्स को जो हार्मफुल है वाटर में. And this is called as flocculation या फिर coagulation. That means if you can see over here, a flock is being created. That means एक heavy material body create हो जाता है जो completely fill होता है किसी भी chemicals या biological products के साथ में, जिसे हम unwanted chemicals या biological product कहते हैं water में. And then the of course upper part of the water is being removed out. This is the clarified water. So अगर आपको ये देखना है करके, so you can just go with one of the best part, that is take a glass of water, and in a glass of water add some amount of a few amount of mud to it, okay, and then try to for a small amount add alum to it, okay, alum, या फिर आप alum का stone होता है तो आप उसे थोड़ी देर के लिए ग्लास में घुमाइए and let it stabilize over there. After a span of time you can find that the water is clear on the upper part, and whatever the pollutant are there, it is there at the lower part of your glass. Now that is called as flocculant, which also takes place at a larger quantity. So you can do this experiment at a smaller quantity at your home also. The next part after pre-chlorination, aeration and coagulation is called as the polyelectrolyte. It is also called as coagulant A. Now what takes place over here? जो coagulant form हो चुका है, इसे जिसे हम thick flock भी कहते हैं, इसे पॉली इलेक्ट्रोलाइट मेथड से रिमूव आउट किया जाता है. दैट मीन्स क्या होता है कि अपर पार्ट ऑफ क्लैरिफाइड वाटर इज रिमूव्ड आउट और इलेक्ट्रोलाइट पास करके इसे और ज्यादा थिकन किया जाता है, जिसकी वजह से क्लैरिफाइड वाटर में ये मिक्स ना हो पाए. ओके, द नेक्स्ट पार्ट इज सेडिमेंटेशन. Now this is for the removal, or I can say separation of the suspended solid particles. Now हमने clarified water remove कर लिया है, but again we are not sure about it because some microbial activities or some chemicals also can get passed with this part also, and henceforth हमें बाद में sedimentation करना पड़ता है. So sedimentation में क्या होता है, जो भी बहुत minuscule particles होते हैं उसे हम फिर से क्या करते हैं, suspend करते हैं, उसका हम फिर से एक बार flock तैयार करते हैं और separation करते हैं, so that again I can get much clarified, purified water. Now when you are doing sedimentation, of course you are trying to use again some chemical components, electrolytes, so that again a heavy material is being settled down which was not earlier settled and then passed on. Then it goes with the process called as desalination. Of course, we know that we are trying to remove out the microbial products, the chemical products which are harmful. But what about the salts? Now this is done with the help of the desalination method. That means desalination में salts का removal होना शुरू हो जाता है, and this process is of course taking place after the filtration process. So you can see the sequence that we are first of all trying to trap. That means हम पहले क्या करते हैं, प्री क्लोरिनेशन के साथ में पहला बायोलॉजिकल ग्रोथ को हैम्पर करते हैं या फिर बंद कर देते हैं. नेक्स्ट एरेशन के साथ में हम हार्मफुल केमिकल जैसे आयरन और मैंगनीज होंगे उसे डिसॉल्व करते हैं, सो दैट जब हम इसमें कोएगुलेशन प्रोसेस डालेंगे, कोएगुलेशन प्रोसेस में इस वाटर को डालेंगे, ये सेटल डाउन हो जाए उस फ्लॉक में. फिर हम कोएगुलेशन करते हैं, दैट मीन्स जो भी अरेस्ट किया था हमने प्री क्लोरिनेशन के साथ में और एरेशन के साथ में, अब वो एक फ्लॉक तैयार हो जाएगा या फिर वाटर बॉडी क्लियर होना शुरू हो जाएगा. नेक्स्ट करते हैं पॉली इलेक्ट्रोलाइट, दैट मीन्स अपर पार्ट ऑफ फिल्ट्रेट को निकालकर हम थिकर पार्ट को रिमूव करना शुरू कर देते हैं. बट अभी भी वाटर बॉडी जो क्लियरिफाइड है वो कंप्लीटली क्लीन नहीं है, इसीलिए हम उसे sedimentation में डालते हैं, so that whatever the minuscule particle is there right now still in your water body is then removed out. Then it goes with filtration process, जिसमें काफी ज्यादा अमाउंट में फिल्टर होके पानी आता है और इसे अब सस्पेंडेड पार्टिकल्स का फ्री माना जाता है. दैट मीन्स कंप्लीटली सस्पेंडेड पार्टिकल फ्री हो जाते हैं इसमें से. एंड फाइनली हम इसमें से सॉल्ट्स रिमूव आउट करना शुरू कर देते हैं. दैट मीन्स हम इसमें से अब ऑलमोस्ट अमाउंट ऑफ क्लियर्ड वाटर देखना शुरू हो जाता है. बट अगेन देयर रिमेन्स अ पार्ट जिसे हम डिसइंफेक्शन कहते हैं. सो डिसइंफेक्शन क्यों किया जाता है, बिकॉज वी नो दैट सम बैक्टीरिया आर प्रेजेंट इन द वाटर राइट नाउ ऑल्सो विच आर हार्मफुल, जिसकी वजह से ऑफकोर्स यू कैन फाइंड दैट इवन दो वी आर फॉलोइंग द BIS स्टैंडर्ड, स्टिल देयर आर वाटर ट्रीटमेंट्स मोर नीडेड, बिकॉज बहुत ज़्यादा वाटर डेवलप्ड डिजीजेस है हमारे कंट्रीज में. लाइक यू कैन फाइंड डायरिया वेन एवर द रेनी वाटर और रेनी सीजन स्टार्ट्स, it gets easily mixed with our water system and henceforth there is a diarrhea or cholera, jaundice. This system occurs frequently, henceforth the disinfection is being continued. But as I said earlier also, there is no perfect solution or there is no perfect process for purifying the method. Treatment always varies, treatment always exceeds. Maybe in some part the treatment has to be followed again and again. That means ये हमने जो steps देखे थे, it has to be followed again and again where you find hard water. So you can just imagine if there is a hard water or saline water, desalination method वहाँ पे काफ़ी ज़्यादा करना पड़ेगा. या फिर अगर कोई ऐसा पार्ट है जहाँ पे काफ़ी ज़्यादा अलगी ग्रोथ है, माइक्रोबियल ग्रोथ है, लाइक किसी पॉन्ड में अगर काफ़ी ज़्यादा अलगी ग्रो हो चुका है तो वहाँ पे प्री क्लोरिनेशन काफ़ी ज़्यादा करना पड़ेगा. या फिर अगर कोई इंडस्ट्री की एरिया से वाटर आ रहा है तो यू नॉट हैव एन आइडिया कि इंडस्ट्री रिमूव्ड वाटर शायद से मिक्स हो गया होगा वाटर में, तो आपको एरेशन मेथड ज़्यादा करना पड़ेगा. सो लाइक आई सेड, यू डू नॉट हैव अ वेरी क्लियर परफेक्ट मेथड ऑफ प्यूरिफाइंग द वाटर. ओनली वी आर गिविंग अ रफ आइडिया ऑफ प्यूरिफिकेशन मेथड. And this completely depends upon the manual system. That means आपको पहले BIS system करना ही होगा. जिसकी वजह से आपको पता चल जाएगा कि what amount of solid contents or impurities are there in the water, and then you have to adapt the complete method. That means which method is needed or which step needs to be done at a more part, which step needs to be done at a lesser part. ये आपको बाद में आइडेंटिफाई होगा जब आप पहले BIS कर लोगे और सिस्टम्स में आपको पता चल जाएगा अमाउंट ऑफ पोल्यूटेंट प्रेजेंट. Of course, the system completely depends upon the operator. Okay, henceforth automation यहाँ पे काम करता है, but at the same time the operator or the complete प्लांट जिसके हाथ में होता है केमिकल प्रॉपर्टी चेक करने के बाद में ही किया जाता है. सो इट इज कम्प्लीटली डिपेंड अपॉन द फैक्टर ऑफ ऑपरेटर्स. सो ये कितना कॉस्ट करेगा, स्किल लगेगा, इट इज कम्प्लीटली अ वेरी डिफरेंट पार्ट, एंड वी आर नॉट गोइंग टू एंटर इन दिस पार्ट बिकॉज वी नो इफ वी एंटर इन टू दिस पार्ट इट इज अ ह्यूज एरिया टू स्टडी, लाइक what quality, what cost, etc. is needed, because automation is one part which is having a huge cost, then purification method is having a huge part. Okay, so you people just have to remember, keep in mind the steps which I have told you right now, and also keep in mind that these steps vary from water to water, that means region of water to water, and it is completely based on locally operator systems. सो ऑटोमेशन भी है, बट लोकली ऑपरेटर सिस्टम्स कैसे है, वाटर स्टैंडर्ड कैसा मेंटेन हुआ है, वाटर स्टैंडर्ड पहले कैसे मेजर हुआ है, कौन से रीजन में मेजर हुआ है, ये सब देख के ही बाद में हम इसका डिसाइड कर सकते हैं कि कौन सी मेथड को ज़्यादा फोकस करना है. सो पीपल, आई होप यू पीपल हैव अंडरस्टूड दिस पार्ट of BIS standard method, and specially you have to keep in mind something with regards to the water treatment with regards to drinking water, also more reference to the surface water supply. Till the next lecture we meet, study hard.